Okay. So I'll take questions. We'll begin with Deep, who has his hand up. Deep, go for it.
Sir, I wanted to ask, how does a cell recognize what is damaged or what is not functioning?
Good question. Good question. Right. That's a important question to ask. So there are many ways to do this, right? And it's fairly. It depends on that particular organelle or tissue. In some cases, there could be certain changes that accrue over a period of time, right? Which means that it's not like it is damaged per se. That is, it is broken in some ways. But there could be changes, for example, in the phosphorylation of a protein, right? And this phosphorylation happens over time, over a period of time, a certain percentage of a certain protein is getting phosphorylated in the cell. And that could be a trigger, right? See, the critical thing here to consider is that there is no, I think, we have a tendency to think that there must be some kind of a sign or a symbol that says, okay, this is the organelle that needs to be kind of sent for processing through the autophagy pathway. It's possible that when these autophagosomes are being formed, that the proteins that are present that are creating these structures may also bind to certain things, preferentially, and one of those could be a change like this to proteins that are present on the surface of an organelle. There could be changes, for example, in the lipid composition that could also contribute to this. To the best of my knowledge, it isn't very clear if there is like a specific trigger for a specific autophagy pathway to be activated. There could be many things that could drive this, right? But it's an important thought to have, saying, what exactly can flag this, right? And in many of these situations, these are changes that accumulate over time, right? I don't know if you've read about telomeres and how telomeres work, and how the age of the cell is determined in some way, right? So if and there is an accruing over every generation, right? Every time the cell divides, there are small changes that happen, and after a point, you kind of know what the health of the cell is based on these telomeres. So similarly, there could be small changes that, for example, a mitochondria could accrue over a period of time. And if you're interested, go look this up. Some of this is actually known in terms of what proteins are modified and could be a trigger to send this to the autophagy pathway. Okay. Vaishnavi, your query.
So why do autophagosomes have a double-layered membrane?
Because they are kind of, they are created within the cell, right? So they also, have a way to kind of, they need to be able to be created, within themselves. And that's why they begin. So it is essentially, if you think about it, it's actually a vesicle that kind of folds and then goes this way, right? And so there are two membranes here, and if you can imagine it that way, right? And that's just the mechanism that they have evolved with. To the best of my knowledge, there isn't a huge advantage to having two membranes. It's not like the membranes that having one bilayer and another bilayer actually does something hugely important here. I could be wrong about this, so look this up. I'll also see if I can find something that says that having two membranes is actually going to be beneficial in some way. To the best of my knowledge, I don't think so. Okay, so let me check and come back to you as well. If I find something, I will send it out to the rest of the class also.
Okay.
Disha, your query.
So what is amphisome and how is it different from phagosome?
What is the?
Amphysome. So it was in the image, one of the images.
Okay, let me go pull this up. So that's essentially the same autophagosome, but now, it has merged with something that is an endocytic component, right? And this is by endocytosis, there is a mechanism called heterophagy, which allows you to pick things up from outside as well, right? And the amphysome is essentially a fusion of both of these, right? So there is a something that's coming from the plasma membrane that fuses with the autophagosome is what creates an amphysome. So these are essentially all structures that are leading to their fusion with the lysosome, causing breakup of whatever components they are carrying, and the fact that they are brought together in this way, they are named differently in some cases. There are a couple of questions on the chat box that I'll take. Sai Chinmay, and then I'll come back to the hands that are up. Sai Chinmay has a query saying, how is the functioning of the lysosome? This is different from the autophagosome. Does the autophagosome also release energy as a result of degradation? Is that how it is different? So the lysosome essentially carries the components for breakup, right? For chewing everything up and digesting everything. The autophagosome, is just capturing the component that needs to be broken down, right? So unless the autophagosome by itself does not have any lytic enzymes. And so the lysosome coming with the autophagosome is what then allows for the autophagosome to digest everything that it carries, right? Kedar is a query. Sorry?
So what does the autophagosome carry?
Autophagosome captures whatever needs to be broken down, right?
I see. Okay.
Right. So it is essentially capturing things that need to be digested, right? Right. Kedar is a query. Can we say that the autophagosome is a kind of lysosome? Sure. See, the thing is, it's not a lysosome simply because it is a vesicular structure. It's not a lysosome because for it to be called a lysosome, it has to have lytic enzymes, right? The coming together of the lysosome with the autophagosome is what allows for the lytic enzymes to be part of it. Endosomes and phagosomes. So endosomes are essentially endocytic vesicles, and we haven't talked about the process of endocytosis yet. Maybe I'll speak a little bit next time about endocytic pathways, because so far we've talked about membranes that are coming in from within the cell, right? And this is also, and the endocytic pathway is essentially a way of pinching membranes from the plasma membrane, right? So the plasma membrane bends and then pinches off. And this is a way of regulating the composition of the plasma membrane. It's a way of regulating the amount of receptors that are present on the plasma membrane. It's a way of taking things in and delivering them inside the cell. All of this is done by endocytic pathways. So that structure that comes in is an endosome, right? And that's the difference between so the difference between an endosome and a phagosome essentially would be that when it comes to taking up, it's also phagocytosis is also an endocytic process, but there the component that is taken in is something which is a food particle or something that is harmful to the cell, like a bacteria that it wants to take care of. Endocytic pathways or endosomes are largely, working to kind of bring in membrane and receptors that are present on the membrane, right? Lysosome is basically vesicle. Lysosome is, yes, a vesicle, but it has a vesicle with a very unique pH and, a whole bunch of enzymes that can chew a bunch of different things off, right? And that's why they are different. Anand has his hand up for a while. Anand, can you go ahead and ask your question?
Yes, sir. Sir, I wanted to ask that are there any appreciable differences between the lysosome content within a species? Like, for example, I wanted to use the Jacob's disease, like the prions, which are actually misfolded proteins, right? So it was seen in some tribals where they, ate the mortal remains of the previous people, and they got the same disease. They like it got infected in the brains. Like there was accumulation of proteins. So it seems that not all people died because of the disease. Like some were able to maybe digest that protein, and that would be via lysosomes, right? So I was thinking, are there any variations in the enzyme content of lysosomes within a species?
That means you and me could have different lytic enzymes.
Yeah, some were able to digest those.
So,
Yeah.
some of that could also come from the kind of exposure that you've had. It could also have to do with other components that are other players that are required for the coming together of these proteins, because they are essentially aggregates, right? So the process of aggregation is could also be influenced by many things, not just one, right? And so the short answer to your query, whether lysosomes, for example, in different cell types could have different lytic compositions, it's possible that the composition of the enzymes that are present here, the ratio of the relative enzymes that are present in lysosomes that are part of a particular organ could be different from another organ. I think the fact that individuals have many different cell types, and the not just the lysosomal content, but other content could also be different between individuals. There could be many factors that influence, the nature of these enzymes. But I'm not sure whether it's known that there could be one or two enzymes that, one person may have that another person may not. Okay. Let me know if you find something. I don't know if that is really the case. Okay. I would not think that might be the case. Right. Varat has a query on the chat box. The Golgi apparatus does the finishing of proteins and makes modifications such as lipid modification or glycosylation of proteins. How does the Golgi know that a particular type of protein is needed in a certain organelle? While making a protein itself, why don't these changes occur? Do all the organelles convey their protein need directly to the Golgi only? So these are very interesting questions, Bharat, right? Let's take one at a time. So does the Golgi know that a particular type of protein is needed in a certain organelle? Probably doesn't. Okay. So I think the Golgi does what it does. And this is something to also kind of wrap our heads around, right? Like, for example, when endocytosis is happening, and this is true for many cellular processes, right? That is triggered by something that drives endocytosis. And it's probably not thinking about a lot of other things that are happening in the cell at that point of time. This trigger has happened. There is something A that has told it that now endocytosis has to happen. It will go and do endocytosis. The stuff gets endocytosed and brought in. Then the availability of components inside, right? What is available? What does it? Is it in proximity to where exactly this endocytosis has happened? Could all be influencers to what happens to this vesicle, right? And the same is true for components from the Golgi, right? The Golgi is taking proteins that are coming from the ER. There are times that the amount of protein synthesis may go up, the number of vesicles are coming in are higher, the crowding of proteins in the Golgi may be high, the time that an enzyme gets to spend with a protein could get affected. So the efficiency with which the Golgi works could change, be high or low. But once the Golgi processes a protein and puts it into a vesicle, then the vesicle just kind of bumbles around, right? If there is a protein, a motor protein that needs to carry that vesicle, that's available immediately there. It goes, captures it, right? Now this motor protein sits and starts walking with it. Now the vesicle has no clue where it is going, right? The motor protein kind of knows, where it's headed, right? In some ways. So a lot of this is not very carefully pre-planned. So it's not like, this protein knew right at the beginning that it needs to go bind to this particular motor protein to go to this compartment. The availability of this motor protein, the fact that, so if there is a protein on this vesicle that can bind this motor protein, there might be proteins on this vesicle that bind 7 other motor proteins too, right? So what decides where this vesicle goes to? The factors that could influence this is, of the 7 motor proteins that it can bind, which of these motor proteins are available there? Maybe 2 are available, right? Now, if 2 are available, what is the relative concentration of these 2, right? Is one present 5 times more than the other, right? The affinity with which they bind, right? The one that is present 5 times more binds with very poor affinity, right? So it will keep binding, falling off, binding, falling off. The one that is less may bind very firmly, right? And that could influence where this vesicle goes to eventually, right? So as much as, there is a way for things to be carried to a particular site, it is influenced by many of these immediate decisions that happen, right? So I don't think the Golgi is thinking, looks at a protein and says, "Arey, this is the protein that ye to apna favorite protein hai." So, I think the Golgi just looks at the protein and says, "Arey, I have a protein, I have to process it. Let's look at what signature it has, um, and take care of it," right? Does the Golgi keep track of how many times it has seen that protein? Probably not, right? Every time it comes, that protein comes, it sees it as new and processes it, right? While making a protein itself, why don't these changes happen? So the mechanism, the machinery, the conditions that are required for the enzymes that work on this, it's possible that there could be a glycosylation enzyme that is sitting right next to the ribosomes. One of the considerations here might have been that the ribosomes are essentially trying to make a protein and have it prepared and sent out, right? And they want to do this as efficiently as possible. Having a lysosomal enzyme sit right next to there will delay a bunch of things, right? Because proteins are being made very rapidly. The glycosylation process is kind of happening at a certain rate. So there is a possibility that keeping them apart allows for things to move at a certain rate and speed here and at a certain rate and speed here. The conditions that are required for these enzymes and the conditions that are required for the ribosomes to function optimally could vary. This could be another reason to kind of keep them apart and separate them as well, right? Do all the organelles convey their protein needs directly to the Golgi? That's not how this works. Okay, so remember, protein, it's highly possible that there is a signal that is a receptor that is bound to the plasma membrane that talks to the nucleus to tell that it should synthesize, activate the transcription of a particular gene that now codes for a protein that now is getting made a lot more, right? And once it gets made, where it gets delivered depends upon that milieu and that environment. So I don't think organelles per se are doing this, right? It may affect the fact that this receptor is bound eventually leads to the delivery to a particular organelle. But did the organelle actually tell the Golgi? I don't think so, right? Does autophagy happen in non-dividing cells too? How are they able to replace the organelles that are lost? So it's a good question. So there is some autophagy that happens in non-dividing cells. I'm not sure whether the rates are very radically different, right? And the synthesis of new organelles, is not tied to division, right? If that's how you're thinking about it, Medha, right? It doesn't have to be that a cell has to divide to make new organelles. So it's possible that once the components are broken down, that new organelles are also made in cells that are not dividing, right? What is the alternate to lysosomes do prokaryotes have? So I'm not sure there is an equivalent of the lysosome in prokaryotes, right? And check this up. Let me know if you find something. But to the best of my knowledge, I don't think there is something that does pretty much the same thing. Now, yeast obviously have these, right? but I'm not sure whether prokaryotes, like bacteria, have a lysosomal equivalent or something that actually breaks this down. Let me go look this up as well, right?
Sir,
And
excuse me. Sorry to interrupt, but
sorry,
even I was having the same doubt.
sorry,
I
you have to
was
identify. One sec, you have to first identify yourself.
okay. So I am Vaishnavi. Even I put that hand up because I was also thinking about the same, but I thought it is a silly doubt. Sir,
There is nothing called a silly doubt. Okay, as long as
but even I was comparing lysosome and amoeba. With that, you showed that slide of processes, right? Endocytosis process and phagocytosis process there, right?
right.
On that slide. So I was also thinking the same. Even amoeba also taking the food in, and also there are vacuoles also therein, and it is
So
also
amoeba are a different thing, right? I don't think, I think when you say prokaryotes, you're thinking about bacteria, right? I don't think you're thinking about paramecium or amoeba, right?
no. I
Uh,
was thinking about unicellular cellular organism,
that's
microorganism.
different. That's not prokaryotes necessarily, right?
Okay.
Prokaryotes, uh, you know, uh, if you're thinking about amoeba and paramecium, they have slightly different mechanisms of handling this.
Yes.
So bacteria per se, whether they have lysosome-like structures, let me look up. I'll find out, and if what I find out, I will let you know.
Okay, sir. Thank you.
Okay. Ayush, last question.
Sir, in the autophagy, like the five-step process, I think it was the Scientific American article picture. So, in that, the first step was like when there's like a signal, and all of the signals were, they didn't have much to do with like the damaged mitochondria itself. So, sir, my question was, would it be right to say that the autophagy is induced by causes which could damage organelles rather than the damaged organisms themselves?
No. Autophagy cannot be. See, the thing here is that the autophagy is part of the natural process of the cell as well. So it's something that keeps happening irrespective of whether there is a damage-inducing agent or not. Now, a damage-inducing agent may increase damage, and therefore cause a need for increased autophagy, right? But in the normal maintenance of the cell, autophagy is important. And so that creation of an autophagosome is probably happening at a steady, constant rate in all cells, right? It doesn't have a lot to eat and process if damage is not a lot. And this is standard, level of processing that it does. But there are situations where there is damage to the cell, and the autophagy process needs to be kind of hastened or sped up. And that's also possible, right? So both scenarios could exist in that sense, right?